Welcome back to the Deep Dive. Today we're tackling something that is arguably one of the darkest, maybe most frustrating mysteries in medicine.
Yeah, it really is.
You know, usually when we talk about cancer, we focus entirely on the tumor, right? The lump, the invader, the thing that grows.
Right, we focus on the mass itself and how to kill it.
Exactly. But today we're talking about the shadow that cancer casts over the entire rest of the body. We're talking about cachexia. And I have to be honest, before reading the research we're diving into today, I always thought cachexia was just, eating less.
Yeah, that's a common misconception.
Like you get sick, you lose your appetite, you look a bit thin.
If only it were that simple. No, cachexia it's a completely different beast. It is not just starvation. It is a fundamental, aggressive rewiring of your metabolism.
Wow.
It's a wasting syndrome where your body literally starts to digest its own muscle and fat regardless of how much you eat. You could be eating thousands of calories and you would still waste away. It accounts for nearly half of all cancer deaths. Yes. Specifically in pancreatic, lung, and gastric cancers, it's often the cachexia that kills the patient before the tumor burden itself does.
That is a staggering number. So today, we are diving into a groundbreaking paper from 2026. It's by Shi and colleagues, published in the journal Cancer Cell. And the title is a bit of a mouthful. It's called Tumor-Immune-Neural Circuit Disrupts Energy Homeostasis in Cancer Cachexia.
It is a dense title for sure, but the story it tells is honestly like a detective novel. It solves a central mystery that has baffled oncologists for decades.
Which is?
How does a relatively small tumor sitting quietly in the pancreas convince the entire body to hit the self-destruct button?
And the answer isn't just, the tumor releases a toxin. It's way more complex. It's like this twisted game of telephone involving three totally different systems in the body.
Correct. It's a triad. We have the tumor, the immune system, and the nervous system. And the scary part is that they are all talking to each other in a continuous, vicious loop.
Okay, let's unpack this. Our mission today, for you listening, is to trace this signal step by step. We're going to detail every single mechanism. We need to figure out who starts the fire, who fans the flames, and why the fire department, which is our brain, ends up making it worse.
I like that analogy. Let's start with the setup. Imagine you walk into a hospital room, you see a patient with advanced pancreatic cancer. They are emaciated, skeletal. This is the clinical reality of cachexia.
Right.
Now, for a long time, we've known about a specific molecule floating in the blood of these patients called GDF15.
GDF15. And that stands for growth and differentiation factor 15, right?
Right. Think of GDF15 as a stress siren. It's a hormone. When the body is in severe distress, GDF15 levels spike. It travels through the blood, hits the brain, and effectively screams, "Emergency! We are sick! Stop eating immediately!"
So it's the appetite killer.
It is, but it's also a metabolic switch flipper. It puts the body into a hypercatabolic state, which means rapid energy burning. Now, here is where the detective story really begins. For years, the assumption in the medical community was simple.
Let me guess. The tumor is stressed, so the tumor makes the GDF15.
Exactly. That was the consensus. The tumor is the bad guy, so the tumor makes the bad hormone.
But this paper throws a massive plot twist at us right in the beginning.
Massive. The researchers use a highly advanced technique called single-cell RNA sequencing.
Which is where they look at the genetic readouts of individual cells.
Precisely. They basically took the tumor environment apart, literally cell by cell, to read the barcodes and see exactly who was writing the genetic instructions for GDF15.
And they found that the tumor cells they were barely making it at all.
Right. The tumor was essentially quiet on the GDF15 front.
Wait, hold on. If the tumor isn't making it, but the patient's blood is absolutely full of it, who is the factory?
The macrophages.
The immune cells.
Specifically, the tumor-associated macrophages. Now, macrophages are immune cells that have infiltrated the tumor. They're supposed to be the frontline soldiers defending us, eating up cellular debris and fighting pathogens.
Right, they're the good guys.
Usually, yes. But instead, inside this tumor microenvironment, they've been corrupted. They've been turned into a factory for this wasting hormone, GDF15.
Okay, this changes everything about how we view cancer's local environment. The tumor isn't holding the weapon. It's convinced the security guard to hold the weapon.
That's a great way to put it.
But how? I mean, the macrophage doesn't just decide to betray the body on its own.
No, it doesn't. And that brings us to the first real leg of our journey. Let's call it the bribe. The tumor secretes a completely different molecule called CSF1. That's colony-stimulating factor 1.
CSF1.
Think of CSF1 as a recruitment poster, or better yet, a malicious reprogramming code. The tumor pumps out CSF1 to call macrophages to the site. It says, hey, come over here, we need you.
And then what happens?
When the macrophages arrive and bathe in that CSF1, they undergo a structural and functional change. They get reprogrammed. And once reprogrammed, they start pumping out GDF15.
Okay, so let me do a quick mini summary here for the audience. So far, we trace the line like this. The tumor releases the bribe, CSF1. The macrophage picks it up. The macrophage then releases the siren, GDF15.
Exactly.
So ask yourself, if your own immune system is the factory causing the wasting, how do you shut it down without destroying the immune system? It's a tricky problem.
It is, but I want to pause here because the paper goes even deeper into the molecular weeds. It asks a fundamental question: how does the tumor know to make CSF1 in the first place?
Right, because if we want to stop this at the absolute source, we need to know who is giving the orders inside the cancer cell itself.
This is where we get into the heavy machinery of the cell, and it's fascinating. By analyzing the genes of aggressive cachexia-inducing tumors, the researchers identified a specific protein that was heavily overexpressed. It's called ZIP4.
ZIP4. Now, I know ZIP stands for Zert and ERT-like protein, which is just a very fancy biochemical way of saying it's a zinc transporter.
Correct. It moves the metal zinc across the cell membrane.
But wait, zinc. Why is a nutrient delivery truck running a genetic factory? Isn't its job just to move metal around for basic cell function?
In healthy tissue, yes. That's the boring utility work. But in pancreatic cancer, metal metabolism gets wildly distorted. ZIP4 gets promoted. It stops being just a delivery truck and starts acting like a factory foreman.
A rogue foreman.
Exactly. It's highly overexpressed in these wasting tumors. And as the foreman, ZIP4 bosses around a specific worker. This worker is a transcription factor called ZFP64.
Okay, let's define transcription factor for everyone listening. That's essentially a protein that runs into the DNA vault, finds a specific gene, and flips a switch to turn that gene on or off, right?
Precisely. It transcribes the DNA into actionable instructions. So here is the chain of command inside the tumor cell. The foreman, ZIP4, yells at the worker, ZFP64.
And what does the worker do?
ZFP64 physically runs to the DNA vault. It searches for the promoter region of the CSF1 gene. Think of the promoter region as the steering wheel for that specific gene. The paper even identified the exact binding spot, a sequence they call the P4 fragment.
That is an incredible level of detail. They found the exact docking bay.
They did. ZFP64 binds tightly to the P4 fragment, turns the CSF1 gene on, and boom, the cell starts churning out the CSF1 bribe at industrial rates.
I love this factory analogy. It makes it so clear. So let's visualize this inside the tumor cell. Foreman ZIP4 orders worker ZFP64 to pull the lever on the CSF1 machine.
Correct.
The CSF1 product gets shipped out of the cell, and that CSF1 floats over to the macrophage, which gets reprogrammed and starts blasting the siren, GDF15.
You've got the first two legs of the triangle perfectly mapped out.
Okay, before we leave the local tumor environment, let's do our first 5-minute recap for everyone tracking along at home. We've established the sender of the signal.
Let's hear the bullets.
Bullet 1, we have Cachexia, the deadly wasting syndrome.
We know the immediate systemic cause is GDF15, the don't-eat hormone. Bullet 3, the big surprise. Macrophages make the GDF15, not the tumor cells.
The tumor forces the macrophages to do it by sending a chemical bribe called CSF1 in bullet 5. The tumor makes that bribe because of a strict chain of command, the ZIP4 foreman activating the ZFP64 worker.
That is a perfect, high-density summary of the local environment. But now we have to zoom out. GDF15 is a hormone. It doesn't stay local. It enters the bloodstream. It goes on a journey.
Destination, the brain.
Specifically the hindbrain, down in the brainstem. This is the primal, ancient control center of the body. It regulates autonomic functions. Now, GDF15 can't just walk into any cell in the brain. It needs a highly specific keyhole.
And the lock waiting for it is a receptor called GFRAL.
GFRAL.
Exactly. GFRAL is the receptor, and it's exclusively found in this specific part of the brain. But it doesn't work alone. It works with a partner, a co-receptor called RET. RET.
So they form a complex.
Yes. When GDF15 from the blood hits this GFRAT complex on the brainstem neurons, it's like pressing a massive red panic button in the headquarters of your body.
And what actually happens when that panic button is pushed? 2 devastating things happen. First, anorexia. The desire to eat completely vanishes. The brain tells the body, no food. But the second thing is arguably much more destructive. and this is where we need to spend a moment. It triggers a profound metabolic shift.
This is the part of the paper that genuinely terrified me. It's the burning the furniture to keep the house warm phase.
That is exactly what it is. To prove this, the researchers looked at the respiratory exchange ratio, or RER, in mice.
Let's break down RER.
RER is a clinical measurement, a ratio of carbon dioxide produced to oxygen consumed. It's a very simple, reliable way to tell what kind of fuel a body is burning at any given moment.
Right, so if the number is high, closer to 1.0, you're burning carbohydrates, like a normal person after eating a big meal.
Exactly. And if the number is low, closer to 0.7, you're primarily burning lipids, you're burning fat.
And what did they see in the cachectic mice?
Their RER was rock bottom. They were in a constant state of aggressive lipid oxidation. They were rapidly incinerating their fat stores even when they weren't moving. But it gets even worse. The paper details a physiological phenomenon called the browning of fat.
Browning, like searing a steak.
No, like turning on a space heater. See, humans have different types of fat. We have white adipose tissue, which is essentially our energy savings account. It just stores calories.
Right.
And we have brown adipose tissue, which is loaded with mitochondria and burns energy to create heat. Babies have a lot of it to keep warm, but adults don't need much.
Okay, so what does GDF-15 do to this fat?
The brain signal triggered by GDF-15 tells the white fat to turn beige or brown. It initiates a process using a protein called UCP1, which literally uncouples the energy chain.
Uncouples it.
It means instead of using the fat to create usable cellular energy, ATP, it just wastes the fat as pure heat. It turns your energy savings account into an uncontrollable furnace.
So the body isn't just using fat for energy because it's starving. It's actively wasting it as heat.
Exactly. The metabolism becomes incredibly inefficient. It becomes an energy vampire.
And what was the genetic proof that GDF-15 is the sole cause of this furnace effect?
They use knockout mice. These are mice genetically engineered to lack the GDF-15 gene entirely.
So they can't make the siren.
Right. And what happened? Even when these knockout mice were implanted with aggressive pancreatic tumors, their RER stayed high. They kept burning carbs normally. They kept their fat mass. They did not turn into furnaces.
That is wild. So the tumor creates a local signal chain that eventually hacks the brain to flip the burn everything switch. So let's do a mini summary here. Imagine being the brain, sitting in the dark skull, getting this false alarm via GDF-15 that the body is under severe threat. What would you do next?
You fight back.
Exactly. And that is where the story turns from straight line into a vicious, inescapable circle. Because the brainstem doesn't just receive the signal and sit there. It reacts.
Sensing this massive emergency, the brain activates the sympathetic nervous system.
The classic fight or flight system.
Yes. The brain thinks the body is under attack, maybe by a massive system-wide infection, so it fires up the sympathetic nerves. Now, these nerves run all over the body, branching out everywhere, including directly into the pancreas.
Including right into the tumor microenvironment?
Yes. The study found that in cachectic patients and mice, the tumor tissue was absolutely flooded with a specific neurotransmitter called norepinephrine. And they found high levels of tyrosine hydroxylase, which is a marker showing dense nerve fibers invading the tumor.
Norepinephrine, that's adrenaline's chemical cousin, right?
Right. The brain is dumping massive amounts of stress fuel directly into the system. And here is the absolute cruelty of this biological circuit, that norepinephrine hits the tumor cells.
And the tumor cells, do they just ignore it?
No, they are primed for it. They have specific receptors for norepinephrine called beta-adrenergic receptors, specifically ADRB2.
Okay, so norepinephrine binds to ADRB2 on the tumor cell.
Yeah.
And what happens when the tumor gets hit with this stress signal from the brain?
It causes the tumor to produce even more ZIP4.
Oh, wait, I see the trap.
Do you see the loop closing?
If the norepinephrine increases ZIP4, and ZIP4 is the hormone that starts the whole process,
then the entire cycle restarts, but stronger.
Let me trace this out because this is the feed-forward loop from hell. Step 1, the brain panics and fires norepinephrine down the nerves. Step 2, it hits the ADRB2 receptor on the tumor. Step 3, the tumor makes more ZIP4 hormone. Step 4, more ZIP4 means more ZFP64 workers making more CSF1 bribes.
Keep going.
Step 5, more CSF1 recruits even more macrophages. Step 6, those macrophages scream out even more GDF-15 sirens. Step 7, the massive wave of GDF-15 hits the brain, and the brain screams even louder, dumping more norepinephrine back onto the tumor.
It is a self-sustaining feed-forward loop. It's exactly like audio feedback at a concert. You know when a microphone gets too close to a speaker?
Yeah, that awful screeching noise that just builds and builds.
The microphone, which is our macrophage, picks up the sound. It sends it to the speaker, the brain. The brain blasts the sound, norepinephrine, back at the microphone, the tumor. And it just gets louder and louder and louder until it's deafening.
And that deafening systemic screech is cachexia. The body is literally screaming itself to death.
Precisely. And the tumor is capitalizing on this chaotic feedback to grow, suppress regular immunity, and disrupt the entire host ecosystem.
That is terrifyingly elegant. Nature can be so ruthless when a mechanism gets hijacked.
It truly is.
Okay, we're about 10 minutes in. We need to solidify this entire triad in our minds because this loop is the absolute core discovery of the Shia paper. Let's recap the full circuit.
Let's do it, leg by leg.
Leg 1, tumor to immune. The tumor uses ZIP4 to make CSF1, bribing the macrophages.
Leg 2, immune-to-neural. The reprogrammed macrophages release GDF15, which travels to the brainstem, binds to GFRAL-RET, and turns on the fat-burning furnace.
Leg 3, neural to tumor. The brain panics, fires the sympathetic fight-or-flight nerves, dumping norepinephrine back onto the tumor, which boosts ZIP4 and restarts the entire cycle.
You've got it perfectly. And understanding this loop explains why just treating one part of cachexia, like just telling a patient to eat more, completely fails. It's a self-sustaining engine. The food just gets burned in the furnace.
But knowing the engine exists and knowing exactly how the gears connect means we can try to throw a wrench into it. And the paper goes into this extensively, right? They didn't just map the disaster, they mapped out how to stop it.
They did. And this is the hope section of our deep dive. They tested breaking the triangle at all three corners using targeted pharmacology, using specific drugs.
Let's walk through the evidence levels for these therapeutics. Corner 1, the immune leg. The bribe. How do we stop it?
They used a drug called axatilimab. It's a monoclonal antibody that physically blocks the CSF1 receptor on the macrophage.
So it puts molecular earplugs on the macrophage.
Exactly. If the macrophages can't hear the CSF1 bribe, they don't get recruited to the tumor and they never get reprogrammed to make GDF15. The result: greatly reduced macrophage infiltration, lower systemic GDF15, and less wasting in the subjects.
Brilliant. Okay, corner 2. The neural signal traveling in the blood. The siren itself.
For this, they used an antibody called ponsegromab. This one is pretty straightforward in its action. It binds directly to the GDF15 molecules floating in the blood and neutralizes them.
Like shooting down a missile in midair.
Yes, it intercepts the siren before it ever reaches the brainstem. The brain stays quiet.
And corner 3, the brain's reception. The lock on the door.
This is really the most fascinating part clinically. They used a drug called selpercatinib. It's a highly selective inhibitor of ARP. Remember, RAT is the co-receptor on the brainstem working with GFRAL.
Right, the panic button.
Yeah.
And what happened when they used selpercatinib in the cachectic mice?
It completely blocked the brainstem response. The mice maintained their body weight. They didn't start burning fat. But here is the fascinating clinical nugget that connects this purely academic paper to real patients in hospitals right now.
I love these connections. What is it?
Selpercatinib is already an FDA-approved drug.
Really? For cachexia?
No, not for cachexia. It's used as a targeted therapy for certain lung and thyroid cancers that are driven by specific RET gene mutations.
Okay, so it's a standard oncology drug for specific tumors.
Right. And for a while, oncologists had noticed a very strange side effect in clinical trials. Patients taking selpercatinib often gain weight.
They gain weight. During active cancer treatment, that's almost unheard of. Usually everybody loses weight.
Exactly. It was an anomalous side effect they couldn't fully explain. Why are these patients getting heavier and preserving muscle? But now, thanks to this 2026 paper, the mystery is solved. We know why.
The drug was accidentally breaking this exact cachexia feedback loop.
Yes. By blocking RET to stop the tumor, it was simultaneously blocking the brain from hearing the GDF15 scream. It turned off the furnace.
That is incredible. It's like finding out a weird side effect is actually a silver bullet cure for a completely different, deadly problem.
It's beautiful science because it validates the entire mechanistic model in humans, not just in engineered mice. The pathway is real.
I want to touch on the evidence a bit more regarding the physical outcomes. You mentioned the treated mice kept their weight, but in cachexia, weight isn't just fat, it's functional muscle. Did they actually keep their strength? Did it improve their quality of life?
They absolutely did. The researchers didn't just put them on a scale. They measured grip strength using specialized force meters. The mice treated with these blockers were significantly stronger.
That's huge.
And they looked at the actual muscle fibers under a microscope. The cross-sectional area of the muscle cells was vastly larger and healthier. And importantly, they stopped that pathological browning of the fat we talked about earlier.
So the UCP1 heater was turned off.
The heater was turned off. The lipid energy remains safely stored as white fat.
This really changes the paradigm of oncology, doesn't it? Instead of just focusing 100% of our effort on kill the tumor at all costs with toxic chemicals, it's shifting to cut the communication lines and protect the host.
Exactly. It's recognizing that the tumor's deadliness isn't just about its physical size, it's about its systemic influence.
So what does this all mean for the future of treatment? We have these drugs, some are even FDA approved. Are we going to see a standardized Cachexia cocktail prescribed alongside chemo?
I think that is highly likely in the near future. We are seeing a major shift where we treat the tumor-host interaction as aggressively as the dividing cancer cells. This paper strongly suggests that if you can use an anti-CSF1R or an anti-GDF15 drug right alongside your standard chemotherapy, you might drastically improve survival outcomes.
Because if they don't waste away, they can handle the chemo.
Exactly. Remember, cachexia often makes patients too frail to even tolerate life-saving chemotherapy. Their bodies just can't take the toxicity.
So if we can stop the wasting, we buy them time. We buy them resilience and strength.
And in oncology, time and strength are everything.
I want to zoom out one last time before we close out this deep dive. It connects back to a really profound evolutionary question.
I know where you're going with this.
Why does this mechanism even exist in our DNA? Why does our body have a built-in, hardwired self-destruct loop? It seems like a massive biological design flaw to have a panic button that burns your own muscles.
That is the million-dollar question in cachexia research. But if you look at it through an evolutionary lens, this mechanism actually makes perfect sense. It looks exactly like the body's natural, ancient response to a severe acute bacterial or viral infection.
Like getting a terrible case of the flu.
Or a systemic blood infection. Think about sickness behavior in animals and humans. When you have a really bad flu, what happens? You immediately lose your appetite, you go to bed. Why? Because digesting food takes a massive amount of energy. And biologically, restricting nutrients like iron and glucose starves the invading bacteria.
That makes sense. And the fat burning.
You get a fever. Why? Because the brain mobilizes energy from fat stores, uncouples that UCP1 protein, and generates heat to literally cook the pathogen out of your body.
So the body is intentionally mobilizing energy from fat to fuel the immune system and raise core temperature.
Exactly. It recruits the immune system macrophages. It suppresses appetite to starve the bug GDF15. It mobilizes fast energy norepinephrine. It is a brilliant, highly coordinated emergency protocol designed for a 3-day or 5-day infection.
But cancer isn't a 3-day infection.
Exactly. Cancer is chronic. The tumor hijacks this brilliant evolutionary button, and it just holds it down. It locks the acute emergency protocol in the on-end position permanently.
And because it's wired as a feed-forward feedback loop, it just gets louder and louder. The cancer uses our own millions of years old defense mechanism to drain us completely dry.
That's the ultimate tragedy of it. The tumor itself isn't doing the heavy lifting of cachexia. It's recruiting you, your own immune cells, and your own nervous system to destroy you.
That is a really heavy thought, but also oddly hopeful.
How so?
Because it means the tumor is entirely reliant on our systems to do the damage, and we know how to manipulate our own systems. We have drugs that calm nerves. We have drugs that modify immune cells. We aren't trying to invent a poison for a totally alien entity.
Precisely. We don't always have to beat the tumor at its own local game. Sometimes we just need to cut the wires. We just need to hang up the phone.
I love that phrase. Hang up the phone.
If we break the circuit, we break the syndrome. The patient regains their strength, and then we can fight the tumor on better terms.
What a phenomenal way to frame it. Before we sign off, I want to throw a final provocative question out to you, the listener. We've spent the last 20 minutes talking about how the brain, the immune system, and the tumor are irrevocably locked in this triangle. If we can block this deadly wasting without ever actually touching the tumor itself, does that change how you fundamentally view cancer?
It's a great question. Is cancer simply a genetic disease of a single mutant cell, or is it a systemic disease of whole body communication?
It forces us to look at the patient as a whole, interconnected ecosystem, not just a carrier of a localized lump.
Absolutely.
Next time you hear about metabolic diseases or even just unexplained weight loss, visualize that triangle we built today. Tumor, macrophage, nerve, the factory foreman, the siren, and the screeching audio feedback loop.
It's a powerful visual memory aid. Once you see the triad, you can't unsee it.
Check the show notes below. We put a detailed diagram of this exact feedback loop there because, honestly, seeing the visual makes all these acronyms click into place. It's a complex circuit, but as we learned today, it is solvable.
It absolutely is.
Thanks for listening. Keep diving deep.
Keep diving.